Good morning, everybody, and welcome to our Rhode Island Innovator series. This series is intended to focus on discovery and innovation in the greater Rhode Island area in the life sciences sector. My name is Carol Malice, and I'm the Executive Director of Rhode Island's Life Sciences Trade Organization and state affiliate for Biotechnology Innovation Organization. And we serve as the voice of the life sciences industry. And we're a member-based organization, and we offer our members access to resources, training, mentoring, business advisory services, and information sharing, such as this morning's webinar. And this morning, we're talking with Ciencia, a company that is transforming cutting-edge innovations into the future of medical diagnostics. And related to their science, they have built the Sentinella platform to improve clinical diagnostics by increasing both accuracy and access. The team, we have the team with us. So we have Arturo Pilar, the president and CEO, Will Page, the senior scientist, and David Potter, vice president of commercial operations. So welcome everybody this morning. Thank you all for our guests for being here. And we're going to start with having each of you tell a little bit about yourself. So maybe Arturo, you can tell us a little bit about who you are and your background.
Sure. Initially, I'd like to thank you, Carol, for putting this all together, and thank you for the Rhode Island bio community for joining in. We really appreciate that. So anyway, my name is Arturo Pilar. I co-founder of Ciencia and been in business since 1992. So I take care of project management, proposal writing, and general management of the whole company. Right now, we want to try and commercialize the technology that we've been working on for several years.
Thank you. Will?
Hi, I'm William Page. I am a practicing internal medicine physician at the Miriam Hospital in Rhode Island. And I am also a long-standing Ciencia employee. Briefly, I started working for Ciencia actually when I was in college, and I continued there with the goal of eventually reaching medical school. I've been working with Arturo for years and years and years, initially doing clerical duties, pushing paper and answering phone calls, and then eventually getting to the point where I became more comfortable with the technology, started helping our chief technology officer write grants, won some grants based on these ideas, and I'm now trying to help with this push for the Sentinella platform to become sort of the future of clinical diagnostics outside of the traditional laboratory testing environments.
Thank you. And David.
Okay, hi to everybody. Many of you know me and vice versa. Basically, my background, I spent the better part of 20 years spinning out life sciences technology innovation from 4 different universities, getting the companies launched, in a couple of cases actually all the way through the companies being sold. In 2017, an old friend and colleague asked me to come to Brown University to be the Senior Director of Technology Transfer, with a special focus on helping gain commercialization traction for promising intellectual property at the university. Spent nearly 4 years there, had some great times, met some just great people, and some great innovations, including one of them, Johnny Page. Hey, Johnny, we're gonna have to touch base soon, and I'd love to catch up on how things are going with what you're doing. But anyway, upon deciding to leave, Johnny introduced me to Will, and we talked. And so what Ciencia is doing is what I wanted to do, again, back as a civilian, of helping promising young technology companies get commercialized, get funding, and get all the pieces lined up to be able to move forward.
Excellent, thank you. So these sessions really focus on 2 areas, the entrepreneurial story and the innovation. So We're going to start with a little bit about the background of the company, the entrepreneurial story. So Arturo, can you tell us, how did the company get started and when?
Sure. In 1992, myself and two professors from the University of Connecticut, we decided to start Ciencia. And one gentleman was a physicist who sadly is no longer with us, but he had some very, very cutting edge research that he felt there was a commercial application for, having to do with spectroscopy. Anyway, we learned about the SBIR program, and we were very fortunate to get funding. And so that was really the way we got started. We got a space together, and we started buying equipment. And yeah, the SBIR program has been key to our success.
Excellent. And then how did you meet Will?
So through a dear family friend, I've known Willie since he was born, really. I've known him a really long time, and it's been great to mentor him. And he's been a really valuable person in our organization.
Yeah, it's one of the, if I may just jump in, it's one of those things where Arturo was the sort of perfect connection, the perfect fit for me, as I had just graduated with a chemistry degree, and I knew I wanted to go into medical school, but I had lacked biological science experience, research experience. I was sort of trying to pad a resume, if you will, and Arturo was the perfect connection for somebody whose parents are both musicians and writers. Here was somebody who knew science. And so I started working at Ciencia then, and it just kind of spiraled. Now we're here.
Yeah, Willie was fantastic in putting together proposals. And we, continued to buy more instrumentation. Now he's actually done some design work. He worked closely with our chief technology officer, who was the former biomedical research individual at the UConn Health Center in Farmington. And so it's been a great collaborative team that we put together. So.
Excellent. And then David, you mentioned that you met Will through Johnny. Do you want to talk a little bit more about how you came to the company?
Well, yes, I actually, when I was still working with faculty members at Brown, like Eric and others, I was introduced to Will and Arturo because at the time they were thinking of going in several directions. So then after I decided to leave Brown, shortly thereafter, I was reconnected. And that was, it'll be a year ago in May, I guess. And ever since been working with them on various aspects, again, of putting the pieces together that we need to advance the ball commercially.
Thank you. And then
Yeah.
Arturo, are there others on the team that you want to talk about?
Yeah. So I mentioned our biomedical engineer, chief technology officer. His name's Ernie Ginion. And he is the PI on most of our projects. He's got several incredible talents: mechanical engineering, electrical engineering, understands physics, and he also helps in writing proposals. We do a lot of collaborations with area universities, one being University of Connecticut and Storrs. And from there are two individuals that we've worked with extensively. One is George Gibson, who is a physicist, and he's the designer of our optical platform. A fantastic collaborator. And then there's Nate Cady from SUNY Poly in Albany, New York. Another individual, I'm sorry, at UConn is Michael Lines, who is an immunologist. We've done several SBIRs and STTRs with him. We have one currently pending and actually that we're working on. The other individual is Nate Cady, as I mentioned before, at SUNY Poly. He is a microbiologist and is involved in science. He develops our chips, which we will get into later. So it's been wonderful working with these individuals, and we have quite a strong team.
Excellent. You mentioned SBIRs. It sounds like that was very important to getting started. So can you tell us a little bit about, it's been quite a long journey for your
Sure.
company. What was it like when you first got started?
Well, we set up a kind of a lean and mean operation. Since then, and that was in 1992, since we've received 39 phase 1s and 21 phase 2s. We've worked with several agencies, starting with DOD, and within DOD, the Army, the Navy. We worked extensively with NASA, DTRA, EPA, NSF, and now primarily NIH. So that would be the primary funding mechanism, or the customer that we're working with now is NIH.
That's a very impressive track record. 39 phase 1s, and did you say 21 phase 2s?
21 phase 2s, yeah.
So tell us your secret sauce.
Well, I would say initially, my former colleague was an incredibly talented grant writer. And we felt that in this whole process, it's really important to understand that there's a market need for the project that you're proposing for. He was extraordinary in terms of our success in getting things going and winning a lot of these proposals.
I would imagine that over time, your business model has changed. So can you take us from where you started to
Sure.
where you are today?
Sure. So when we initially started with funding from DOD, we were working on hyperspectral sensors. And so that took us into, we always wanted to get into biomedical sciences. That was what we really wanted to do. We started working with NASA, and we started developing detection technology. So with the goal of doing that, we did a lot of work with NASA, then NSF, and NIH finally. So this current platform that we're going to be discussing today, we've been working on for several years, and we've managed to raise approximately $10 million non-dilutive funding from NIH.
Yeah, and I will say there's, and Arturo, maybe you were going to get into this. There was kind of a confluence of events that occurred, about 10 years or so ago now. First of all, with the death of Sal, who was the company's first president. That was around the same time that we were investigating the earliest aspects of what has become our Centinela technology. And, several years later, we've had a good overlap between a technology, a market need, and then, the company's values of trying to put together, trying to move away from projects for the Army, as prescient as they may be on this inauspicious day, and also actually work towards medical diagnostics.
Right. I may add that we also had commercial contracts, many of them, and the platform that we're discussing now came out of initially PerkinElmer. And they hired us to look at a technology with the idea of setting up another company to commercialize that. At the end of the day, we did our job, and we thought it was a fairly complicated instrument, and we had many ideas on how to simplify it and make it cheaper. Unfortunately, PerkinElmer wasn't interested in that at that point or going in that direction. So we just took off with it and focused on this technology and started getting a lot of funding. I may add that that technology that we helped develop ultimately got bought out by GE Medical, and then Biacor, a company in Sweden, has now purchased that.
So let's talk about the current technology. I know it's a fascinating technology, and please take us through the development of that.
Yeah, so maybe I'll jump in here.
Yeah.
Just because I've it has been an interesting path for us, and there have been a number of I don't want to say false starts, but a number of ways in which we thought we were going to go, and then for any number of reasons found ourselves now where we are today. So the PerkinElmer technology that Arturo was talking about is a grating coupled approach to surface plasmon resonance. So for those who are familiar with surface plasmon resonance, briefly, it is a technology that allows light energy to go into a surface under appropriate conditions. And if those conditions are met, that light energy does not reflect back off. It basically gets sucked into the surface. That has been used primarily for, the biggest applications are certain medical research, but a lot in drug discovery research. It's probably the biggest market for surface plasmon resonance at present. PerkinElmer was exploring a grating coupled approach to surface plasmon resonance, which briefly is a way to do a somewhat complicated set of assays in an inexpensive, rapid, easily manufactured, easy to use kind of way. And that's actually where we started. We started with this phenomenon that had been previously described, that PerkinElmer wanted to explore but didn't really want to spend the resources on themselves. And that is ultimately the technology that was spun off to get to GE Healthcare via Biacor. And our twist on it is that we found out through some in-house experimentation that the light energy, as it was being absorbed into the plasmon mode, we could do something useful with it. So instead of shining the light on the surface and seeing is it absorbed and just basically changing delicate parameters to see how the assay responds, what we found is that we can use that light energy to essentially funnel energy into fluorophores, into fluorescent molecules. And this is a way of basically performing commonly used biological assays in a way that greatly reduces the amount of material and therefore greatly reduces the cost. So over the years, we have worked to demonstrate that we can perform assays like ELISAs, like Western blots, like ELISpots, and even like certain flow cytometry assays using less than a nanogram of the costly reagents. And we print those costly reagents on little chips that we are presently manufacturing for a couple of dollars a piece, and we could be manufacturing for a couple of cents a piece. Our sensor chips are based on a technology very similar to the manufacture of compact discs or DVDs. And so, in short, our approach as we see it right now is to try and pursue both research and clinical diagnostics in a way that makes things cheaper and faster and better. And we've been doing that for really the better part of a decade, initially through some our earliest efforts had this fluorescent enhancement only as sort of an ancillary feature, but we have now worked to focus on this, which greatly reduces our instrument cost, our per unit assay cost, and there are a number of other features and advantages that we can get into. I do want to share a slide here, if I could, Carol, basically that just kind of, I think, gives a good visual of what we're able to do. So give me one moment. All right, is everybody able to see my screen?
Yeah.
Yes.
Okay. So our patented technology, we're calling the platform the Centinela platform, but this is grating coupled fluorescent plasmonics is sort of the scientific name for it. And I like this image because I think of it as just a clear demonstration of the power of the system. So what you're seeing here is you are seeing a single sensor chip where half of it has our pattern, the half on the left, and half of it is just flat, plain, simple gold. Everything else is the same with the exception of our pattern, which again is manufactured in a very similar way to compact discs or DVDs. On the left half, there are 8 identical reagent spots. On the right half, there are another 8 identical, the same 8 identical spots. So a total of 16 spots. And I think it's clear to see 8 of them are visible, 8 of them are not. And that is the enhancements that we offer. There are many people, I'll just note, there are many people out there who are trying to look for the most sensitive technology to be able to detect one particle in a quadrillion, detect one atom from halfway across the room. There are certain advantages to those kinds of technologies, primarily in research markets. But when you're getting into clinical relevance, it turns out that biology is messy. Clinical medicine is messy. And so we have elected not to pursue the lowest possible limits of detection, although we're pretty good. We have instead decided to reduce our assay costs. So each of these spots that you see here is less than a nanogram of material, less than 4,000ths of a cent for a typical antigen. And it allows us to print complicated assays with abundant positive and negative internal controls on a chip, and with a reagent cost that's still less than $1. So I'm going to stop sharing, and I think that's sort of the introduction.
This might be a good time to ask anyone here if they have any questions about this technology. Anyone who is listening in this morning. I guess not.
Oh, maybe I'll
Yes.
volunteer kind of a
Okay.
starting point to that discussion.
Sure.
Now, as we all know, timing is such a critical factor in whether and how innovative technology catches the attention of funding sources and the marketplace, et cetera. And I'll just say that some of you are probably aware of the whole Theranos fiasco going back 5-6 years ago, where claims were made about miraculous diagnostic capabilities that have since been radically debunked. And I would say that some of the after effects of that debunking had created some barriers of, it makes it even more, you have to prove it to me, when it comes to new diagnostic technologies. On the other hand, timing is great right now because the pandemic experience has exposed how the more traditional model of people going to a hospital or doctor's office to get tested to see if they have an infectious disease, and then maybe 3 days later, they hear back from their physician, no or yes, and here's what you do or you don't do about it. So there is both a public health, a consumer, and other pushes to make high quality inexpensive diagnostics more available to consumers at convenient places that quickly give the answers to physicians as to what the condition is, what this means for treatment. So our capability here is in motion at a time that responds to those environmental opportunities. On the other hand, what we're talking about is disruptive in many ways of the long established business model of a lot of big companies who see what the future is coming and don't want to miss that. On the other hand, they're making an awful lot of money right now from the way they're doing things. And so there's this yin and yang about how fast and how aggressively they want to pursue doing what we're doing. So we're in that kind of period right now of navigating, how to find our initial, relationships commercially. But again, that the timing and external forces piece are, so important in the entire equation.
Sure. So are you getting actual pushback because of the Theranos story? Has that affected you in any way, or what seems to be feedback you're getting?
Yeah, maybe I'll pick up on that. The current pitch, as we're putting it forward, is not dissimilar to the now I believe it's accurate to say documented fraudulence that was being perpetrated. Nonetheless, there is a real desire from these markets. There is also a fair amount of appropriately placed skepticism from those who are trying to review these kinds of innovations. So I will say that it's a common point of comparison. I guess I wouldn't say that we're getting a ton of pushback, because I think that we can distinguish ourselves clearly and effectively. To that end, we are doing two things, I think, that are very different from what Elizabeth Holmes did. One is that we're not claiming that we do everything. We simply aren't. We have a lot of assays that our system is not well suited for. This is not the platform you want if you are measuring your blood sodium level. This is not the platform you want if you are measuring the size of your red blood cells. However, there are laboratory devices on the market now. The iStat series is a common one that simply meet those needs, but where they fail is the slightly more exotic but still very common tests. So, for example, things like Lyme disease testing, things like autoimmune hepatitis testing, things that I, as a practicing clinician, order every day, but don't necessarily order on every patient. And so the way that we distinguish ourselves is, one, by only claiming that we perform assays that either we have clearly documented or have good theoretical justification, and of course, saying where we're at with either case. But then two, we're transparent. We show our patents, we show our publications, we are peer reviewed in quality journals. We're showing the work. And so the goal, the goal here is to try and capture the enthusiasm that propelled Holmes where she went, while actually taking into account scientific reality. And so I'm gonna share one more slide that is, I'm not gonna go into all the details here. It's a busy slide, but this comes from one of our papers. This was one that our collaborator did pretty early on in COVID. And this was actually before we got involved with variant testing, but I like the way this graphic sort of summarizes the kinds of things that we do. So just quickly, there are a total of 8 pictures of 8 patients' samples on 8 of our chips. Each of those chips have been printed with the reagents in the key. And essentially, what you can see is that we were testing for, we had positive controls, we had negative controls, we had proteins from influenza, we have proteins from the first SARS, we have a variety of different proteins from COVID. And we were, by printing these chips, each spot that you see there is, again, less than a nanogram of material. Each of these 8 chips, we were able to distinguish patients who had COVID from patients who didn't, patients who had had the flu in the past from patients who didn't, and make real valid clinical decisions based on good statistical modeling and methods that allowed us to be predictive about if somebody had COVID or not. I'm gonna leave this slide, but you know, we as a company are working to broaden or to demonstrate the breadth of our potential use case. And so we've explored COVID, we explored Lyme disease. With Lyme disease, we actually demonstrated using CDC validated samples. We demonstrated an improved sensitivity and the same 100% specificity with Western blot and ELISA, the standard methods. So our assay actually, N is equal to, I believe, 28 at this point, because this was an unfunded study, but you know, with a well-defined CDC sample set, we actually demonstrated, you know, significantly superior results. And so, yeah, so I guess to touch back on Theranos, you know, I think that they have been a valuable lesson in importance of not over-promising. And they also have been a good frame of reference as we seek to define ourselves in the market space.
Excellent. And for those of you who haven't read Bad Blood, The Secrets and Lies in a Silicon Valley Startup by John Carreyrou, it tells the whole Theranos story. It's an excellent read. And I'm sure we'll be seeing a movie at some point, but I highly recommend it if you want to become familiar with that story. And in speaking of that, they had a relationship with Walgreens. So do you, Arturo, Will, or David, do you see you having something like that in the future?
Well, if I may, and I apologize, I don't mean to sort of dominate here, but I'm going to share one more just because I have the slide that sort of illustrates one of the avenues that we could approach, and this is sort of what David was talking about before too. So I am often faced with the limitations of our healthcare system. I am often faced with social determinants of health, where patients simply can't get a ride to go to the doctor or to the pharmacy or whatever else. And so there are the sort of humanistic elements, but there's also the business sense of, if people don't have to take 8 trips to different locations, they'll probably be happier and more likely to engage with your services. So one of the places that we anticipate putting these kinds of tests would be Walgreens, CVS, a pharmacy, where we can make a comparison between a standard approach where if I'm a patient and I have a new problem, I call up, I make an appointment, or I walk into a walk-in clinic. Eventually I'm seen by a provider. The provider orders blood tests. I then go to a phlebotomist. I return home, I get a call maybe saying, oh, you know, we got one test, but it was kind of equivocal. We need to do a different test. I come back in, or I come back in to discuss the results, the concerning results of a test. And then the physician prescribes prescriptions. I go to my pharmacy now, I pick up the prescriptions, and finally I'm able to start treatment. You know, depending on how efficient you are, you could say that there are 8 one-way trips involved in, you know, but by the time you leave your house and by the time you return home with prescriptions in hand. We aim to improve efficiency at pretty much a large number of steps. But should we be integrated into Walgreens or CVS or somebody, you know, of that nature, a patient could go to the Minute Clinic. They could have their finger stick tested based on information that they entered in. I have been having a cough, or, you know, tell the person doing triage. The appropriate tests could be listed for given concerns or complaints in advance. Finger stick testing could get you could allow you to collect the sample efficiently. Those tests can be running at the same time that you have your encounter with the provider. Just as a quick side note, our tests are at present about 30 minutes start to finish, although I think we have some time that we can shave off there. But importantly, the preparation of the test occurs outside the instrument, which means that our overall throughput, these can be performed in parallel and just read in the machine in 2-3 seconds. So potentially thousands of such assays could be performed in a day. But going back, this information is now available to the provider in that encounter, allowing the decision to be made in that moment to prescribe as necessary. If you happen to be in a CVS Minute Clinic, you get to retain that patient's business. They are there for the provider encounter. Why not fill the prescriptions just down aisle 3? Pick them up and walk home with them. You go there, all your healthcare needs are met, you return home.
And Johnny, if I, Will, if I could pick up just a second on why that's important too. The good news is that there are other I'm going to call them self-enclosed systems like ours that also could be operated in a mini clinic, which is good because we've already seen these companies are getting funding, they're getting trial utilization, et cetera. The real key thing is, though, as we've gotten to know more about what's inside those systems, we're more and more encouraged that unique aspects of what we have to offer in terms of sensitivity on the science side, cost reduction, and a number of other value propositions that our composite offering stands to be seen as very unique, and will be the source of attraction to commercial customers. And we're just in the process now of designing a strategy for funding to further develop the intellectual property, as well as to more aggressively market the technology. So sometimes it's better not to be totally unique, but distinctly different in a kind of a proven space in the evolving marketplace.
So you mentioned funding, and you mentioned understanding the environment in which you might operate. What do you need to do to get to that next place?
David, you want to take it, or?
Well, I think it's a couple things, Carol. I'd say that as Will mentioned earlier, the different indications or disease conditions that we can confidently prove to have scientific efficacy to produce high quality results. In a couple of cases, there's published proof. In other cases, as Will mentioned, there's a lot of leading indicators which would say with a little more specific development, we would be able to prove out definitively that all those other conditions would be applicable as well. So one of the things we're looking to do is get additional funding support to further prove out the broadly agnostic condition treatment capability. And part two is a collaborative organization that would be, willing to help actually, put some finished units in a test site type of environment where people come in and so forth. And prior to that, we'll be getting, regulatory clearance to do this, because there is the regulatory piece as well.
Yeah,
That's
which is not,
Yeah.
I mean, I think that, Carol, I think it does depend how far out are we looking, right? And I think that there is still a ways to go before we are actually in every minute clinic in the country, right?
Right.
And so, the sort of the reasonable bites, the reasonable milestones, I think, include, I think the regulatory hurdle is a big one. The way that the regulatory pathway is set up is that for technologies like ours, where where we use the same reagents that other assays do, you kind of just need a beachhead case. You kind of need a first assay. And for right now, Lyme disease is what's leading. We do have the opportunity to gather a few more samples and, towards COVID, but to be honest, I'm just not sure that that's the best market for us to be pursuing at the moment. So I think Lyme disease is one that we already have an assay that has demonstrated some clinical superiority, at least again in a relatively small sample size. And we would work to build that out as we also work to refine our prototypes and sort of streamline ease of use. To that end, we are very interested in working with Zymedica, who has a longstanding track record of facilitating regulatory approval, as well as the human factors engineering, et cetera, that they do so well that would be necessary to get our instruments into people's hands. So there are stages. I think it makes sense to, I do think it makes sense to continue to build out our claims of other, to prove that we can do assays beyond Lyme disease and COVID. But for those who have experience with ELISAs and ELI spots and Western blot, the reagent selection is very much what defines the assay. And we have been successful with a broad class of reagents in a variety of different applications.
I think
To that end, let me add that we should be hearing very shortly about two large proposals that we have submitted. We expect to hear within a month or so. Again, one of them is an SBIR, another one is a grant with New York State, and also involving Zymedica. So we're continuing to, as well as we'd like to find partnerships with other researchers, we're continuing to write proposals as well.
And actually, one more thing I didn't mention, but Arturo, we are also in talks about. Although I did suggest Lyme, there's some very compelling data sort of hot off the presses with another colleague who's exploring the multi-inflammatory system in children, MIS-C, which is a rare inflammatory condition that children get after COVID for unclear reasons. But we do have collaborators who have been working on a biomarker signature for this disease with the goal of establishing a clinical diagnostic. And without going into details, there have been some encouraging results.
Yeah, I think it's also important to mention that from a business model standpoint, the decision was to really focus on what we do best, which is develop innovative science. That our model will be to license out to the manufacturing, license to entities that do the marketing and the delivery. Right. We're not, our model isn't to become a multifaceted, diagnostic. You got manufacturing, kind of organization. We want to be carefully rooted in again what we know we can do.
And to that, right, and to that end, being able to say that the instrument is FDA approved would be very attractive as we go and we talk to people. However, if there is a potential partner who has their own set of preferences as to what we pursue next, we would certainly be listening.
And you mentioned SBIR funding, but are you looking for non-government resources as well?
Yes.
And
Yep.
how are you going about that?
A lot of cold calls.
Well,
You know,
yeah. Well,
Meetings such as these.
yeah, we haven't formally launched that yet, Carol, but that's in our planning to supplement the non-dilutive funding with some additional, private funding at a certain point.
Sure.
part of it is trying to align our next steps. It would be great as we pursue private funding to have interested parties who would be on the receiving end of this technology, to have them in place with ideally with their preferred milestones and metrics for our instrument. And so we're having those calls as well.
Excellent. Excellent. Well, I'm going to shift gears a little bit because we've talked a lot about the technology, the innovation, the market, the business model for the company. Let's talk a little bit about some lessons learned along the way. So every entrepreneur is interested in knowing some of the challenge. We talked about some of the challenges. You had a death that, along the way. And Arturo, maybe you can address that.
Sure.
What are
Sure.
some of the
Well,
lessons learned in this company?
Initially, defining the market that you want to work with is something that was not our primary focus at first. We would write proposals, we'd win them. I think that is key to a success. The team that you put together is very important. And with respect to proposals, one really important aspect is to have letters of collaborations from people that really say, okay, this technology is fantastic. We support your efforts. If successful, we'd like to work with you further. We found earlier on, we didn't focus that much on that aspect of things. Now we have, and it's made a huge difference in being successful in getting these proposals. One of the problems, well, I wouldn't call it problems, one of the challenges are the wait times between phase 1 funding, phase 2 funding, and we've been fortunate in that we had several projects going at once, and also being able to fill in gaps with commercial projects as well that kept us going.
Yeah.
The three of you are very experienced in your respective areas. What advice would you give to other people who might be starting up a company, funding a company, looking for a particular scientific competitive advantage?
Well, I just wanted to, I think this kind of fits in this question, but also to the last question. One thing that was a lesson learned for me, I, of course, was lucky enough to sort of get brought into a company with an established infrastructure, with, all kinds of PowerPoints that help simplify making an SBIR budget, with, there've been a lot of, there were a lot of things that were very helpful, templates for verbiage about the different components that need to go into the proposals. But two things that I would share. One is that it's important to follow the instructions to the letter. And we really, truly have seen people who come to us for advice who have a topic in mind that doesn't actually have a home in the request for proposals. And that can be absolutely fine as long as you explicitly get the blessing from the program director. But a lot of times people are, they'll say, oh, this part isn't as important. We see that a lot with the commercialization plan in phase 2, that it's almost an afterthought, and it's not. And it's very important that that it is addressed, that it's gone through with the care that that they're looking for. And I guess so. My other point, which is also, I guess, kind of related, but is for me, one of the things that was most surprising about this experience has been, I guess, what I'm going to call the field of dreams fallacy. That if you build it, they will come. And it's not the case with commercialization, at least not in our experience, that you have the technology, you prove the technology, but there really needs to be some legwork. And bringing David on has been very helpful in recognizing that and in reframing our discussion towards commercial products.
I just amplify that a little bit by saying in prior experiences I've had, there's sometimes a tendency to look at first at patents to see if what you're thinking of doing is already patented, and if it's not, it's, oh, well, that's cool. Then we have a kind of a clear pathway. And secondly, along with that, do what I call thorough market research to really be able to objectively determine that there is a commercial need and opportunity to be taken, to be moved forward. And all to be done very early on, because I've been party to experiences where 2 to 3 years worth of development and new innovation, all of a sudden it's, oh my God, somebody else is doing the same thing a little differently, but better. And realistically, there's just not a play. So I know that sounds simplistic, but those are critical fundamental aspects in your thinking about moving forward.
I may add, in the evolution of the SBIR program, I mean, we started early on in the 90s. What's really changed now is, as Willie mentioned, the commercialization plan, which really has to be well thought out and discussed. Even on phase 1, it's important to go ahead and mention that.
Yeah.
It's absolutely key. And the other thing is to get, as I mentioned before, letters of support from people in that established market to support your effort. Absolutely key. Absolutely key.
Yeah. So our proposals will often include, so we will have letters from each of the individual collaborators. We'll have letters from any of the consortium contractual arrangements with, so for subawards to a university, for consulting arrangements with our consultants. But we will often include, so for example, we'll include a letter from Zymetica that says, although, phase 1 budgets are small, we would love to work with you as early on as possible to and work towards a phase 2 or beyond arrangement that shows that we are taking steps toward commercialization, that it's not just one more scientific research project. It's a project that where we demonstrate the fact that there is real commercial potential, and that we have thought about the steps that are needed to take to get there.
To that end, too, it's key to try and have letters of support from potential financial investors as well.
Yeah,
that's really important.
So potential customers, potential investors who could supplement phase 2 funding, any aspects that are considered, higher risk, or any reagents that need to be sort of clarified or validated, any equipment. I mean, we will often, in a phase 1, I think our most recent phase 1, we probably submitted 8 letters
Yeah.
along with the proposal. I mean, it hasn't been funded yet, so maybe this is not maybe the strategy will not pan out. But we will often try and get external validation from as many aspects of this as we possibly can.
So that's a great lead into our final question, which is sometimes we like to give the opportunity to make an ask to people who are listening in this morning. So what would your big ask be for what Ciencia might need related to help or support or resources going forward?
So it's a couple things. We're looking for partnerships out there so that we can continue doing our research, refining our instrumentation. We're also, again, looking for funding to accomplish the same things. Again, we're going to continue doing SBIRs and proposal writing, but we want to try and move this technology a little quicker than the phases that you have to wait for that kind of fund.
And I think if I may just pick up on that, this is a public facing discussion, and our goal here is to continue conversations and to bring on those who can help us, really. We think we're doing something that matters. We think we're doing something that has commercial viability, but also, real potential clinical importance. And so we are looking to people with expertise in these fields, both from a technical standpoint and also commercial standpoint, and also also potentially an investment standpoint. And so we're doing what we can to make this a reality, and we would love to speak to anybody who can help to that end.
Excellent. Do we have any questions at this point from anyone on the call?
Could I? I'm sorry to interrupt, Carol, but what I'd like to do, since this will be up on YouTube, is I just want to flash on the screen just a couple of slides that are reference material for anybody who would be interested in sort of fact checking or getting in touch with us. And so just quickly, so hi, these are our key personnel, or sort of key scientific personnel. A list of our collaborators, people we are working very close with, people who own some of our instruments. In front of us in that picture is one of our earliest prototypes. The instrument is a lot less busy than that now. Here are just some selected publications that support the claims we've made. Also include some of the images I've shown. There have been a couple others that outline sort of similar results, so I decided not to sort of bombard you, and that's it. Our website has patents, it has contact information, and certainly, Carol, although I don't want to necessarily put my cell phone on YouTube, things can be distributed as appropriate to interested parties.
Sure, thank you. And as he said, it will be we will put the recording on our website, and you can certainly reach out to me as well if you need any more information. My email is on our website, so it's another way to get in touch. But before we close, just wanted to see if there's anyone on the call that might have a comment or a question for our team. Eric.
I'll ask a question for you guys. So, as mentioned by Arturo and David times in terms of the market being really important. And so I was just curious, I might have missed this, but you settled on sort of Lyme disease as one of the main things to look at. And I'm looking at this model in terms of having it at, say, a CVS or Walgreens or whatever. Why choose something like Lyme disease, which doesn't seem nearly as prevalent in terms of the testing as something like STDs or something that might be a little bit more commonly used?
Yeah, I can speak to that. Our play is breadth. So our assay is to try, like our goal is to try and actually get ourselves in a position where there is one instrument with multiple tests on a shelf. You kind of grab the appropriate one for a clinical environment. Lyme disease is perhaps not, and this is your point is very valid and something that we have discussed. It is perhaps not the biggest, best play to this end. It is, however, where we have really excellent results to date. And so the goal is to get the instrument approved. There are well-characterized sample sets for STDs as well, I'm sure, but they're well-characterized sample sets for Lyme disease, and it's a bit of a fraught market. The tests just aren't that great right now. So if we can continue to demonstrate clinical superiority, if we can do so in a way that's faster and cheaper and better, I think it's a viable market. There are certainly others, yeah, there are certainly others, but it's one that is problematic. It's one that we've demonstrated we can do better, and it's one with a pretty clearly defined path towards regulatory approval.
Yeah, and kind of marketplace lingo, it is a beachhead demonstration of credibility and differentiation, essentially, that you build from that credibility footprint further onward from there. It sounds awfully simplistic, but for a young company, having that in your repertoire opens a lot of doors to broader conversations.
And it's also $1 billion a year market. Like yeah. It's not enormous, but it's not trivial either.
Right.
We'd be happy to have a small component of that pie. especially as a foot in the door.
Yeah.
And while I'm at it, one of the advantages, so I showed that our sensor is able to detect multiple things from the same sample at the same time. Tick-borne illnesses also are, they often live in the same host, and a tick bite can lead to a variety of clinical syndromes that are essentially identical until you do the tests. So, unless you have, full-blown babesiosis, a lot of times people just have kind of like a fever and they were bit by a tick recently and things like that. So it is an opportunity for us to further differentiate ourselves, should it seem appropriate, by a broader tick-borne panel, which is something we're actively working on and would be pursuing in this phase 1.
Excellent. Any last comments, questions?
I have a question. It was a great presentation, guys. I was just wondering, I know you mentioned the model of sort of a testing for acute illness in an urgent care setting or in the pharmacy setting. And I'm wondering whether you think that this technology could also be utilized either in chronic disease or in screening. I think about neurodegenerative disease a lot, and one of the big challenges there is that detecting it and knowing who's going to develop it and when. And so I guess I'm wondering if, obviously a big limitation is biomarkers, but I'm wondering if the low cost and the high content nature of your technology would be amenable to kind of routine testing as well as more acute setting testing.
So I'm going to answer that, if I may, by actually raising a point that I did want to address earlier too. A huge part of what it means to be a doctor is determining who to test and when. And I think one of the real limitations of home-based testing, of being able to go out and buy 100 COVID test kits, is that if you're testing the wrong population, you increase the likelihood that a positive result is a false positive. Or if you're testing the wrong population, you increase the possibility that a negative test is a false negative. And so in clinical medicine, this is what you talk about as a pre-test probability. And then, you do your test, and based on that, you have a post-test probability. So the answer is that this could be incredibly valuable for a well-selected disease. For something like Alzheimer's, there is a lot of work that needs to be done to identify the right biomarkers. It does seem to be a multifactorial disease, meaning that it may not be one biomarker. Our system is beautifully set up to look at 24 different biomarkers in triplicate, with positive and negative controls, like high quality sort of statistical validity of the test and of the biomarkers. And if you have a good assay, we can do it. It does come down to, are the patients being selected properly? Are the tests being ordered at appropriate times? And so the answer is, it totally could happen. Our platform could be great for it, but we have to be careful about over-promising until we see the data.
Thank you. Thank you. This was an excellent presentation. I want to thank Arturo and Will and David and all of you that joined us this morning. Great to see you all. Please, if you haven't gone to our website, please go to our homepage, ri-bio.org, to sign up for our newsletter so that you can receive more information about events like this one for the future, for the dates for those events, and also as a way to see the recordings of this event and others as well. And if you're not a Rhode Island Bio member yet, we would love to have you join our organization and become a member. We provide seminars and webinars like this morning's. We provide training to our members, as well as significant discounts on some vendor relationships we have related to equipment, lab supplies, insurance, and travel. So would love to have you as a member if you are not just yet. But thank you all again, and I look forward to seeing you at our next Rhode Island Innovator series. Stay tuned for the date for that, and anything else will be on our website. So be well, everyone, and thank you again.
Thank you
so
very
much,
much for
Carol.
everybody for showing up. Appreciate your time.
Thank you.
Thank you.
Bye-bye.
Thank you.